It is my great pleasure to welcome Jeffrey Malins as our speaker today. Jeff received his bachelor's degree in molecular biology and genetics at the University of Guelph in Canada, and then he went on to do a PhD in neuroscience at the University of Western Ontario, again in Canada. He then moved on to the US, came to do a postdoctoral degree, postdoctoral he was a postdoctoral associate at the Haskins Laboratories in New Haven, uh, and subsequently joined the Department of Pediatrics at Yale. And at 2019, he joined the Georgia State Department of Psychology, and he's associated with several different um, institutes there, including the Trans Center, which is the Center for Translational Neuroscience there. Uh, Jeff is doing really interesting work on development and language, and I believe he's going to be telling us a lot about that work today. So, Jeff, uh, thank you for accepting our invitation and looking forward to it.
Thank you so much. Thank you for that kind introduction, and thank you to all of you for attending. And it's really an honor for me to speak to you all today. And I'm looking forward to our conversations and getting to know the Georgia Tech community a little bit better. Unfortunately, with the timing of my arrival and the pandemic and everything, I really haven't had an opportunity to interact all that much with you. And so I'm looking forward to sharing some of the work that we've been doing and some new studies we're trying to get off the ground now that we're sort of back and able to start testing again. So today I'm going to be talking about the brain networks that support word learning and processing. And I like to start with an image such as this one here, which is sort of what inspires a lot of the work that we do, which is thinking about children in the classroom and the skills that are really required to thrive in this kind of environment. And so I'll be talking about some work with children, and I'll also be talking a little bit later on about work that we're currently doing with adults. But our aim in the lab is really to bring that back, to think about how to translate the knowledge that we're gaining about these systems in the brain and how we can actually impact classroom practice as a result. And so today we'll spend the bulk of the talk looking into spoken language processing, which is essentially something that's going on, that as I'm speaking, that you as listeners are taking incoming auditory information, mapping that onto the knowledge of language that you have, and ultimately getting at the underlying meaning of what I'm saying. And this is something that children, have quite a lot of experience with in the first few years of life prior to the onset of reading development. And so a lot of what we look at in terms of reading is how that builds upon or how that scaffolds upon the brain networks that are involved in spoken language processing. And, in that sort of classroom picture, there's a lot going on, and certainly attentional control matters quite a lot for spoken language and for reading, and I think its role is being increasingly appreciated. It's something that we're starting to look at in a few different projects in the lab. It's something I'm happy to answer questions on later. We won't be spending too much time on it today, but it's certainly something that we are interested in. But what I hope this figure also is illustrating is that there's these intersections between these different areas. And that's also something that we're really interested in. And so something we're going to be talking about today is reading and spoken language processing and the overlap between the two. And so the first project that I'm going to talk about is one that we did quite a few years ago now, actually during my time as a graduate student working with Mark Janice at the University of Western Ontario. And it looked at something called phonological similarity. And so this is something that is, as I mentioned, sort of that right now that you as listeners are taking incoming auditory information and mapping it onto your knowledge of language. And what's thought is that when we sort of see something like this, that not only do we activate the spoken language representation associated with that, so cone in this case, but that many other representations are also activated if they share some sort of sound relationship with what we expect. So our predicting brain is constantly trying to figure out sort of this incoming auditory information, what specific words are we hearing, to be able to aid us in getting at the underlying meaning of what's being said. And so when we see a picture like this, a representation for the matching item, cone, the sort of linguistic representation in our brain becomes active. But so do a number of other representations of items that share a certain extent of sound overlap with the expected or target word. So words like cone, for example, and comb in English. And so these are words that are not identical to the expected word, but they're similar. And this would be something like if you think of like, when you type in a Google search bar and you sort of type in the first couple of letters, and it will try and predict what it thinks you're trying to search. This would be something like that are sort of, we have this pool of competitors that simultaneously become active, and then that gets whittled down as we receive more and more information to identify the actual word that we're hearing. So if we hear the sort of the word or the sounds co at the beginning of a word, we would have a pool of competitors active that would include cone, comb, code, and so on, and then sort of narrow that down as we receive more information. And what's been found, in addition to that, and this might be somewhat unexpected, is that not only do we activate or partially activate items that share some onset relationship with what we expect, but we also activate items that share rhyme relationships with expected words. So these would be words like bone and phone, sharing that those word final, what are called phonemes or sounds that overlap with cone. And so in this particular study, we worked with children who were either typically developing or children with a learning disability called developmental language disorder. So this is referred to as DLD, and it's a significant difficulty with learning and understanding and processing spoken language. And it affects about 7.5% of children. So out of a classroom of 30 children, that's going to be about 2 kids. And despite its prevalence, it's actually not all that well known. It's sort of about as prevalent as dyslexia, but likely sort of the people in this audience may not have heard of it. If you don't sort of work directly in the field of language, it hasn't really reached the level of attention that, for example, dyslexia has. And as a result, there's a lot of advocacy by a number of groups. For example, this RADLD group, Raising Awareness of Developmental Language Disorder, to sort of put it on the radar of teachers, for example, that if children are in the classroom and are struggling with understanding instructions, if they're struggling with conversational skills, learning new vocabulary, that DLD could be potentially the origin of that. And so this is a learning disability. And so like other learning disabilities, it's quite specific to a particular domain. In this case, oral language. It can also be observed in sign as well, but today we're going to be talking about oral language. And so like other learning disabilities, it is not associated with sort of, or not related to visual or auditory or motor processing difficulties, nor is it an intellectual disability. It is really specific to the domain of oral language. And often children with DLD will score in age expected levels for other domains related to their education, such as math, for example. There can be co-occurring difficulties with language and with math, and often a difficulty with language will lead to difficulties with math or with reading, as we'll talk about today, but not always. It's thought that there's some separability there. And so our question in this study was whether children with DLD would show differences from their typically developing peers and how they handle this phonological competition. So items like cone and comb that share this similarity in sounds, sort of figuring out what is the actual word that's being said and sort of whittling down that competitor pool. And so to look at this, we used event-related potentials, or ERPs, which I'm sure many of you here are familiar with. So children wore these montages of electrodes, which recorded electric activity passively from the surface of the scalp. And these, we looked at different ERP components that were time locked to the onset of presentation of spoken words. So for example, the word cone when seeing a picture of a cone. And we were interested in components of the ERP waveform related to spoken language processing and related to the resolution of competition between similarly sounding items. So the actual task that the children did is a picture word matching task in which they saw a picture of an item like a cake, and subsequent to that they heard an auditory word over the headphones, and that word either matched their expectations, so they saw a cake and heard cake, or mismatched it, like they saw cake and heard lake. And their task was just to indicate via button press whether or not what they heard matched what they saw. So they're just making a match-mismatch judgment on each trial. And importantly, the manipulation here was that in half the trials, they heard what they expected. They saw cone and heard cone, or whatever item the picture happened to be named. And then the other half of trials, they heard a mismatching word. So either what's called a cohort mismatch with that word the onset shared between cone and comb, a rhyme mismatch like cone and bone, or completely unrelated words like cone and fox, which did not share any phonemes at all or any sounds at all with the expected word. That was sort of our control here. And so what do we find? So this was in a group first of typically developing children. This is the waveform for matching words, time locked to the onset of the spoken word. And here's the waveform for unrelated words. So words like fox when seeing a picture of a cone. And you can see here there's this broad modulation here of a component called the N400. So a negativity around 400 milliseconds. This is associated with that mismatch in sounds from what they expect and also in meaning because the word mismatches from the semantic expectation set up by that picture. And we were particularly interested in a later window of this N400 component called the late N400. And so if we now look at rhyming words, what we saw is that there was an attenuation there. In that compared to unrelated words, there was a reduction of this late N400. So it's still showing sort of a more negative going waveform compared to matching words in that, it is not the correct word, it's mismatching. But that late overlap in sounds between bone and cone actually leads to a reduction of this N400 compared to completely unrelated words, which we think is a marker to sort of sensitivity to this phonological relationship and perhaps a facilitation of processing these items when they actually do come in. And so now if we look at the children with DLD, what we observed, first of all, this is for unrelated words. Again, quite a robust N400 effect. But for the rhyming words, within this late N400 window, there was pretty much no reduction at all. These waveforms are pretty much indistinguishable from each other, suggesting that children with DLD were really treating these rhyming words as if they were completely unrelated. So that cone-bone sound relationship was really doing nothing different than an unrelated word was in terms of sort of facilitating recognition, that it sort of was being treated as if it was unrelated. And so based on this, our interpretation was that this lack of reduction of this N400 for rhyming words, perhaps it's a lack of sensitivity to these rhyming relationships, or perhaps it's sort of a lack of sort of reduction of this competitor pool or whittling down that pool based on rhyme relationships. So it's sort of leading to competition effects that are as great as they are for unrelated words. A bit hard to tell from this, but we think it's sort of something to do with this this rhyme relationship that we observed. One thing I should note as well before I move on is that sort of to connect to the literature, that DLD has often been called a specific language impairment or language impairment in other studies, especially in the past. It sort of moved to this definition of DLD. But if those of you in the audience are familiar with SLI, that might be something that you've heard of before. We're treating it as synonymous here. All right, so that was a taste of some of the work that we do looking at spoken language processing, looking at processing of words that are sound similar or are phonologically similar. And as I mentioned at the beginning of the talk, we're really interested in this relationship between spoken language and reading. And because word reading really builds upon spoken language and the brain networks that support spoken language processing. And in addition, we're interested in the intersection in difficulties between spoken language processing and difficulties with word reading. And so reading disability, or RD, is a learning disability specifically impacting reading, in this case, fluent and accurate reading. It's often sort of treated as synonymous with developmental dyslexia, which is really a difficulty identifying or pronouncing words. or a difficulty with what's called decoding words. Reading disability is a bit of a broader term that sort of specifies a number of difficulties with reading, for example, difficulty with comprehension. Here, we're really focused on word reading difficulties, so this can really be thought of as dyslexia. And it affects about 7 to 10% of children, depending on how it's defined, which, as I mentioned, is about as prevalent as DLD is. And importantly, often co-occurs with DLD. So that because sort of spoken language is so critical for subsequent reading development, that difficulty with developing sort of stable or robust representations for spoken language can then impact subsequent reading development as well. And so often this co-occurrence is observed between RD and DLD. And so our question here was, how do reading disability and then comorbid DLD and reading disability impact how children learn and remember words with different relationships? So here we were looking at word learning, which is a little bit different than the previous study that I just talked about, which looked at spoken language processing. So that was for words that the children already knew, like cake and lake. We sort of selected words that we thought, and we tested this as well, that children at this age level would be familiar with. They were quite simple, often monosyllabic words, so single syllables. But importantly with that, it's still a confound in that type of study because children differ in their familiarity and exposure to different vocabulary items. And so if they're, not as familiar with a word like lake, that's going to impact the ERP waves that you see based on sort of that expectation, the strength of that expectation, and then subsequent violation of it and detection of that. And so in this study, what we looked at was how children learned brand new words. So they were sort of all starting at a level playing field in terms of having no familiarity with any of these items because they're completely made up novel words, or what are called pseudo words, which is that they don't exist in the English language, but they could. And that's because they have sounds that are sort of put together in such a way that they follow the rules of how sounds are put together in English. So they are fully pronounceable words, like peeboo and peebo. They just happen not to exist. And so we had children learn a set of 8 different words. And these words share different relationships with each other within the set. So there were items such as peeboo and peebo, which share that onset information like comb and cone do. So they're cohort items. Rhyming items like peeboo and deeboo, and then completely unrelated items like peeboo and tupa, which don't share any sounds at all. So they would be like that cone fox manipulation from earlier, which don't share any overlap. Again, sort of looking at a baseline of learning these unrelated items. Before I move on, I just want to make sure if there's any questions at this time. I'm happy to answer questions to help clarify things.
Yeah, looks like no questions yet.
Okay, great. Thank you. All right, so in this particular study, this was done as part of a large program project that was led by Robin Morris at Georgia State. And it was looking at reading development in children, specifically within 3rd and 4th grade. So this was done across a number of schools within the city of Atlanta. Children were identified by their teachers as struggling readers. And they participated in a program in which they received in-school instruction in a particular intervention for reading disability. So this was 70 hours of the phonological and strategy training program, in which children learn different strategies to help them decode words that they were not familiar with. So sort of take these longer words, break them down in their constituents, and sort of use words that they do know to help them decode these words that they're unfamiliar with. And what we were interested in is types of measures that would help predict the children that go on to show the greatest gains from this program, and then those that might also sort of change as a result of the intervention itself. So what is this intervention actually doing in terms of the brain networks that support reading and language? And how can we use these measures to help predict the children that are sort of going to benefit most from these programs? And, with an eye to sort of developing measures that can also help predict the children that show the least amount of gains from this type of program. And, could we find a program or design one that's better suited for them and sort of where they are in their learning and brain development? So this is, quite a large program project. Many individuals were involved with this. And importantly, what we did is we collected a number of assessments looking at various aspects of reading, such as comprehension and fluency before and after the intervention, as well as other related skills, such as cognitive and behavioral attention, for example. And we did some neuroimaging measures. We also did some experimental measures. And one of them happened to be this word learning task. So the data I'm going to be talking about today were from the children prior to intervention. And I apologize, this figure is a little bit blurry, but what we did is we had the children complete this word learning task in which they were presented with pictures of unusual animals. So these are animals such as an okapi, for example, in this, which is a little bit hard to see. These are animals that exist, but children in an American context are likely unfamiliar with these animals. And so we associated them with these spoken labels of these novel words like peeboo and peebo. And so children on each trial would see two of these animals, and they would hear a spoken cue like find the peeboo, and they would be asked to click on either the left or the right that matches that spoken label. And importantly, at the beginning, they don't know that either one could be the peeboo. They just guess, and they receive feedback on every single trial. So either that's right, that's the peeboo, or try again, in which they would repeatedly try until they select the correct animal that matches that label, and then move on to the next trial. And so we tracked children's learning of these items over time. They did 6 learning blocks, which took about 20 minutes for them to do. And this was done in schools. And then what we did is we also evaluated their retention of these items on the subsequent day. So we had the children 24 hours later complete a similar task with the same items to be able to evaluate how well they remembered what they learned the day before. And so what did we find? So what's shown here are learning trajectories for the cohort rhyme and unrelated items. 4 on the top is a group of typically developing children. In the middle, children with reading disability, but not developmental language disorder. And then in the bottom plot here, children with both reading disability and developmental language disorder. And so if we look in the top plot here, the black curve is unrelated words like peeboo and tupa. You can see that the children get better over time, so they reach sort of about over 90% accuracy in the final block. But they show quite a bit of a lag for these cohort items in red and these rhyme items in blue. And they do reach similar levels of accuracy by the end of the task, but they sort of take longer to get there. That there's a more difficulty or lower accuracy for these preceding blocks. Oh, sorry. And that was really expected, especially the cohort items like peeboo and peebo. They're quite difficult for children to sort of remember which one's which and keep track of this because they're so similar. So there is an improvement over time, but it is there's a sort of lower accuracy here for the cohorts. But what's interesting is that for rhyme, shown in blue, there is also a reduction in accuracy in the typically developing children. And if you recall back to the ERP study I showed at the beginning that had those words like cone and bone, the typically developing kids showed that reduction of that N400 for the rhyming words that the children with DLD didn't show, suggesting that sort of a marker of typical development is this sensitivity to rhymes. And in this kind of task, that can actually lead to some difficulty, where sort of that sensitivity to peeboo and deboo makes it a little bit more difficult initially to figure out which one's which. So it's sort of, perhaps an unexpected effect, but when you kind of think about it in terms of the literature, it makes sense. And so that also helps explain that why the children with RD here in the black curve shown for unrelated and the blue curve for rhymes actually don't really show much of a difference. That our test here showed that there was, I believe the difference was quite minimal and it was not statistically significant between these two curves. Suggesting that the rhyme learning for the children with reading disability was quite similar to unrelated words, which we interpreted as sort of this lack of sensitivity to rhyme. In addition, what we found is that the cohort words, not only were they sort of lag behind the other two, but they never really got there in terms of their accuracy, that these cohort items persisted in being difficult in a way that they did not for the typically developing children, suggesting that these cohort items were also quite difficult for these children. But importantly, the unrelated items were not, so that the black curve here actually is quite similar to the one here for the typically developing kids. And they sort of get to a fairly similar level of accuracy. And our interpretation here was that it was really the items that share this phonological relationship in terms of the cohorts and rhymes that were specifically difficult for children with reading disability, but the unrelated item, sort of the learning trajectories were quite similar to typically developing children. And now if we look in the bottom plot, what we observed here is that the children with reading disability and DLD, they showed difficulty for the cohorts and rhymes, but they also showed for the unrelated that this trajectory was dampened quite a bit and did not reach the same level of accuracy as it did for the children with only reading disability, which we think indicates that DLD, sort of perhaps one of the origins of that, is a more sort of global impairment that sort of across the board is affecting word learning, not just items that share these similar sound relationships. And so what happened on the next day? Well, this is when we had the children back in and we saw how well they remembered what they learned before, the day before. What we observed is that there was sort of this graded relationship between the three groups, which is that the typically developing children sort of had the highest proportion of accuracy of items learned the previous day, even when we regressed these on their day one learning scores, sort of how well they had learned items the previous day, that there was a higher accuracy of retention for the typically developing children, a lesser one for children with only reading disability, and again for the children with both reading disability and DLD, sort of what we think is a more global impairment that really impacted all word types that was more severe than it was for just the children with reading disability only. And so our interpretation here is that in children with reading disability, we think it's really the items that have that phonological similarity, the cohorts and rhymes that are impacted in terms of both learning and also how well children remember those items subsequently after a period of sleep and consolidation when they've come back the next day. For the fact that the children with DLD as well, that it might be a more broader impairment. And so where are we going with all this? Well, what we really want to do is to bring this back to the brain. So to think about the brain networks that support word learning and how they can lead to the types of effects that we observe behaviorally. And so this model put forth by Davis and Gaskell, which is one really based on sort of complementary learning systems involved in learning and consolidation, suggests that not only medial temporal lobe structures such as the hippocampus are involved in initial encoding of words and sort of that transfer of items into cortex, but that there's this sort of dichotomy between a more dorsal route involved in lexical phonology, so phonology being the sound relationships between different words, like what we observed in terms of the cohorts and rhymes, and then a more ventral pathway involved in more semantic aspects of words, which is more the meaning of these different items. And so what we're interested in is sort of the contribution of these different dorsal and ventral regions in terms of word learning. And so in a study that I'm going to tell you about, we're interested specifically in the supramarginal gyrus located within this dorsal pathway in the parietal lobe, and then the middle temporal gyrus here within this more ventral pathway, both in the left hemisphere, which is sort of the more predominant one for language and word learning. And to examine this, we're going to use repetitive TMS or transcranial magnetic stimulation. So this is a study that we're going to do at CABI. This is a picture here of CABI staff that some of you may know, using the TMS machine there, in which this is a coil that is placed upon an individual's scalp, and that induces has a magnetic field that induces a current that can essentially either excite or inhibit a particular brain region based on the protocol that is used. And so what we're going to do in this study is stimulate either the supramarginal gyrus in that dorsal pathway or the middle temporal gyrus in that ventral pathway. And in particular, we're going to look at what stimulation of the left dorsal versus ventral stream, how that impacts learning and remembering new words. And we're going to use a type of repetitive TMS called continuous theta burst stimulation. And this is really an inhibitory stimulation. We're essentially impacting processing within these regions for a period of time, transiently, And we're going to observe during that time, we're going to have individuals learn these new words and see how their word learning is impacted by that TMS prior to that word learning. And so this is done in collaboration with Dr. Nikki Arrington at CABI. And this is a seed grant that we received that is currently ongoing from the GSU Language and Literacy Initiative. And here what we're going to do is work with 24 neurotypical adults between the ages of 18 and 30, 12 of whom will receive continuous theta burst stimulation to the dorsal stream, specifically supramarginal gyrus in the left hemisphere, and then 12 to the ventral stream, which the middle temporal gyrus in the left hemisphere. And this is a 2-week design that's quite complicated. So what we're going to do is initially in their first visits, participants will receive a baseline MRI scan. This is used for neuronavigation for the actual subsequent TMS. But also they will receive a number of other protocols. So we're going to administer DTI, as well as resting state and task fMRI. We have a couple of reading and language tasks that I'm happy to talk about later. And then what will happen is that participants will receive either active CTBS or sham CTBS, and then learn a set of novel words using that word learning task that I described earlier, after which we will then measure their ERPs using this picture word matching task. And so that's the design there, that in week 1, they have their initial MRI scan, they receive active or sham CTBS, learn the items in the behavioral paradigm. We examine their brain waves using this ERP processing paradigm, and then 24 hours later measure the retention. And then in week 2, repeat the protocol, but they would receive either active or sham, whatever they didn't receive in week 1, and we'll obviously counterbalance that. And then they'll learn a second list of items, similar in structure to the first list, but associated with a brand new set of pictures, a new set of words, and then we'll again have the ERP task and again the retention task afterwards. So again, apologies for this blurry image, but the paradigm is pretty much exactly the same as what we did before. The only thing that we're swapping out here is we're using the noun database for pictures of unusual objects as opposed to unusual animals. And so our participants will learn a set of 8 items, and they'll receive feedback on every single trial, and we'll track their learning over time. And similarly, there will be cohort, rhyme, and unrelated items embedded within this closed set. And then after that, they will perform an ERP matching task, picture word matching task based on the set of items that they just learned. So if they just learned that the Pibu was this strange blue object here, then subsequently in the ERP task, they would receive these pictures of these objects one at a time. And here, spoken words that either match or mismatch the label that they've learned is associated with that picture and make these matching judgments. And here is something a little bit different than sort of the previous tasks. We're curious about how the sort of prior TMS will impact the word learning performance. Here now we're sort of interested in those ERP components indexing sort of the strength of the phonological representation and sort of, that these expectancies of, how sort of hearing Pibu when seeing this item, for example, if that sort of leads to a very strong and robust expectation, then when that gets violated by subsequent input in the mismatch trials, we would expect to see sort of strong N400 effects there. And if we don't, or if we see differences, that's sort of an indication of the strength with which these items got encoded in the first place and sort of the within the brain. And so our hypothesis here is that we think that this dorsal stream inhibition, so this inhibition of the SMG, with the TMS will specifically impact those phonologically similar items. So like the cohort and rhymes, which is really mimicking the children with RD in the behavioral study that only show difficulty with these items, but not the unrelated ones. Compared to the ventral stream in MTG, which we think will lead to a more global deficit across all item types, sort of, linking meaning and sound and sort of the more general processes that support that. And so this is more like the children with both RD and DLD. We think that this MTG sort of, temporary lesion from the TMS will sort of mimic what's going on in the children with the comorbid RD and DLD. And so if that's the case, we first predict that there will be a slower growth in accuracy for cohorts for those with dorsal stream inhibition, as well as this sort of lack of rhyme interference. So it's sort of this competitive effect that we would typically see that we would not see in the case of dorsal stream inhibition. And then when the individuals complete this retention test on the second day, poor retention of those items because of sort of this lack of initial robust encoding in the first place. And subsequently in the ERP task, we think that this will change those N400 magnitudes, such as they're decreased for cohorts, and also that rhymes don't show sort of that characteristic reductions compared to unrelated that they typically would in typically developing individuals. And then for those with the ventral stream inhibition, we think that not only are cohort and rhymes going to be impacted, but also unrelated items. So across the board, learning and retention deficits in all three item types, and decreased N400 magnitudes for unrelated items as well, which sort of are, in a way, is our control condition. We think those two will also show decreased magnitudes, suggesting that it's not just the phonological similarity, but sort of more domain general processing supporting word learning that is impacted by this MTG stimulation or inhibition in this case. And so where's this all going? Well, I started the talk with speaking about two studies that we've done with children, and then ended with this ongoing TMS study in which we're working with neurotypical adults. Part of the rationale for that is really with sort of where the TMS protocols are in terms of working with children, and that we'd really like to establish this effect first in adults before then moving to work with younger populations. But we have submitted an R21 proposal in which we plan to look at word learning and processing in older youth with reading disabilities. So specifically, we're interested in what inhibition of this left dorsal stream will do in terms of word learning. And in the study that I just mentioned to you, the sort of ongoing one that we're doing right now with neurotypical adults, our hypothesis there is that there will be a negative impact on word learning by inhibiting either the dorsal or ventral stream in different ways. But what we're really interested in is sort of this provocative idea, which is that perhaps it may actually aid individuals with reading difficulties. And part of the reason for this is that it's been suggested that individuals with reading disability may have heightened levels of cortical excitability. And one of the findings with TMS is that it's thought to lower cortical excitability with this inhibitory protocol. And so it could be that this rTMS, that administration of that, actually may lower cortical excitability such that it is closer to an optimal range and sort of more ideal for developing these phonological representations in this word learning task. And if that's the case, that could really open the door for thinking about therapeutic possibilities for individuals with persistent reading impairments. So often older youth with reading disabilities are children who have went through intervention programs such as the one that I described earlier, and they haven't worked for these children. And so, these reading difficulties have really persisted for a number of years. And so this administration of rTMS could be something that could sort of be used to prime the system for optimal vocabulary development in this case, which may aid in reading. And in addition to that, we're also really curious about individual differences. So I mentioned that sort of within TMS that there's some thought that perhaps TMS is lowering cortical excitability. There's also some debate about the extent to which that's really the case, or sort of what the exact mechanism is there. And what we're particularly interested in is, can we use different neuroimaging measures, in this case fMRI bold signal, to be able to predict those that show sort of the greatest impact of rTMS on their subsequent learning and retention performance. And so we have these fMRI reading and language tests that we're going to have the participants do in their baseline MRI session to be able to use these measures to predict who will sort of go on to change the most, or sort of have the most neuromodulatory impact as a result of that rTMS. So sort of in the remaining minutes that I have, I'll just introduce one of these tasks, which is one that we developed called the FAST localizer. FAST is really meant to indicate the rapidity of how quickly these items are presented on the screen. So In this task, individuals are presented with either printed or spoken words. Here, we're just going to talk about the printed words. And these sort of flash on the screen rapidly one at a time, and they're in sets of 4. So a participant would see like clay, lawn, flea, vase, where each would flash for up to 350 milliseconds. So quite rapid. And importantly, the relationship between these different items is manipulated on the different trials. So sometimes they are completely unrelated in spelling, sound, and meaning. And sometimes they share different relationships. And so one of the important relationships that we built in here was between words that share orthography, that's what this O stands for, which is spelling, and then phonology, which is sound. So words like boat, coat, goat, float, which all share this OAT word ending. And so all these words rhyme with one another. They're what are called consistent. There's a consistent mapping between spelling and sound. Compared to words like host, cost, most, frost, which also share that OST word body spelling, but importantly are not all pronounced in the same consistent way. So these would be what are called inconsistent words in terms of their spelling to sound mappings. And we're particularly interested in regions that differentiate between these two. And when we piloted this task in a group of skilled neurotypical adult readers, what we found is that in particular, the left supramarginal gyrus differentiated between these items that share the spelling sound relationships versus those that don't. So the words that did not consistently rhyme showed greater activation in this region than the words that did. Within the supramarginal gyrus in the left hemisphere, which happens to be the exact same region that we're planning to stimulate within the TMS study. It's part of our rationale for why we've selected this region and why we think it's sort of critical for these phonological relationships like rhymes. So activation within this task is one of the measures that we can use to predict those that will go on to change the most. And the other measure we're really interested in is not activation, but variability. And so this is a measure that we've been working on the past couple of years, in which we're looking at bold activation and how that actually changes over time from trial to trial, and the fluctuations in this measure across time. So what's actually plotted in this sort of a complex figure, which is kind of meant to be sort of an illustration of the complexity of this, but each of the lines in the different colors is an individual child. And this is across trials within an fMRI task that you can see that the bold signal is sort of bouncing around between these activation levels here. The point being that it could be that the same region shows the same level of bold activation in terms of sort of mean activation across trials, which is often the sort of traditional measurement in task fMRI, but may not show the same variability, that there may be more trial-to-trial fluctuation in one child versus another. And we're really interested in this intra-individual variability in activation. And because I mentioned earlier that there's sort of this thought that TMS may alter cortical excitability, this could be one of the very measures that we may see sort of being predictive of those neuromodulatory effects. So I won't go through it here in sort of the interest of time, but it is one that we have found is related to reading in children within the 3rd and 4th grade age range. And so we think as a result that because it sort of shows some relationship with reading, here in this case it's in the inferior frontal gyrus, but nevertheless we think this measure could be one to look at and could give us some informative information for thinking about these neuromodulatory changes. And so as I mentioned, we're sort of doing this work currently with adults, but we're really sort of, as we sort of narrow down the focus of these regions and look at the impact of this neuromodulation, we can begin to think about how that might actually inform what we think about intervention for reading disability or spoken language difficulty in children. And so with that, I'd really like to acknowledge the many individuals who have participated in all of this work. Certainly Robin Morris and Nikki Arrington, who I've worked with on all of the studies that I presented today, at least the one since my graduate training. And, the many individuals who sort of, worked with all of the children that were tested in this experiment. As you can imagine, doing a reading intervention study with children scanning before and after was a huge undertaking. And so a lot of individuals really did a lot of work, parents and teachers and researchers with this. And I'd also like to acknowledge the members of my lab, as well as collaborators at Haskins and other institutions, and NIH for their support of this work. And finally, thank you to all of you for your attention today.
All right, wonderful. Thank you so much. Here's some applause from myself and everybody else. All right, so let's open it up for questions. If you have any questions, either you can raise hand or put them in the chat. In the meantime, I'll start off with a question myself. So, thinking about reading disabilities in children, I was wondering to what extent is it known? To what extent are these driven by a genetic component, and I imagine that there will be a substantial genetic component, and if there is, you know, has is it known what genes are responsible, and if any genes are known, then is it known what kind of effect they have on the brain? Um, there's a lot of if-thens in that question.
Yeah, thanks for that question. And I actually, when I was at Yale, I was in a reading and genetics lab, and I'll try and sort of say this as best I can with the caveat that this is not really an area that I'm an expert in, but there are certain alleles that have been associated with reading development. In particular, they tend to be sort of regulatory sequences. And one that we were really interested in is one called READ1, which is sort of part of the gene DCDC2. Also, there's a gene called KIA-L319 that's also been associated with reading development. They tend to be more associated with sort of neural migration during very early brain development. They're not really reading, but processes that we think may lead to the development of sort of connectivity of networks that are important for
Mhm.
reading. And so one study that we did was working with children with specific comprehension difficulties in reading. So a little bit different than dyslexia. It's more sort of the children with comprehension difficulties are able to read aloud words sort of individually, but it's more sort of putting them together and understanding sentences and passages that's sort of more the difficulty. And there we saw variants of this READ1 associated with the connectivity between different brain regions involved in executive function, which we sort of think is very especially critical for reading comprehension. So I think it sort of has more to do with the connectivity, but we're definitely curious that variability measure that I sort of finished with talking about. That's one of the areas that we're really curious about is how that might be impacted. So, I'll say that the sort of proportion of variance in reading that's accounted for by genetic factors is quite low, and it's likely polygenic, and it's certainly very complex, and there's a lot of environmental factors that are really going to contribute to this. So but it's not negligible, is what I'll say as well. So there is sort of a real interest in also the predictive utility of these markers as well, because what we know about intervention is that the earlier the children get into the programs, the better they do. And so, often these markers we can use well before children learn how to read and sort of administer these programs as early as we can.
Thank you. All right, so we have a question in the chat. I'm going to read it for you. This is from Robert Liu. And Robert, if you want to follow up, or do you want to read your question? I saw you turn on your video. I think you're muted right now. Can you unmute? Are you not able to unmute, or? Oh yeah, there you go.
Can you hear me?
Yep, we can hear now.
Okay. Right. I think there must be just some long delay. I was curious about the CTBS TMS stimulation that you do. So that's supposed to inhibit, and I was wondering that your protocol seems to have that happen before your learning task, and how long does the induced inhibition last? What's known about that? And why not do it during the learning task?
Right. Thank you for that. Yes. And so we have some pilot data suggesting that it's up to an hour. Sort of that's what's been found in the literature. We had participants complete a word reading fMRI task immediately after TMS, and we looked at sort of the impact of the stimulation on these reading and language networks in the brain. And we found that CTBS, at least in terms of bold activation, even 50 minutes out still impacted. So we had them do this task at 20 minutes and then again at 50 minutes. And by we, it's really Nikki Arrington, who I should give all the credit to for this work. She did this pilot work, and so we saw that at least by 50 minutes there was still an impact. So we think it's sort of long enough for within that 20-minute window of doing the behavioral task that it should still be impacted by that.
I see. And how long do you actually do the CTBS stimulation for?
It's about 3 minutes in length. And yeah, one thing that we're debating, which is sort of something that in the literature you'll see sometimes, which is that like having the participants actually sort of prime the network. So if we have them like say aloud a bunch of rhyming words, for example, whether that might sort of lead to some additional impact of this neuromodulation specifically within those networks.
Okay, thank you.
And if I may follow up on that actually, so how is the IRB going for doing that with kids?
Yes, excellent question. And that's why we're not there. I think we really need to establish these effects first with adults and know sort of exactly what we're expecting to see in kids and sort of this is a little bit exploratory still at this point, really seeing what the impact on this task is going to be. We do think this task is a good candidate for this kind of neuromodulation because the behavioral patterns seem so robust and so clear that we thought this would be a good one to use to really identify the impact of the neuromodulation. But, you know, as I mentioned, we're sort of doing this with neurotypical adults first. We then plan to move to working with older youth in sort of the 16 to 24 age range. And then after that, kind of move into working with the more pediatric samples. But that's really down the road. And there are protocols in place in terms of safety, as I'm sure many of you know more than I, in terms of working with children and administering TMS. But we're sort of, you know, we really want to be further ahead than we are before we go there.
Yeah, that makes sense. Yeah, I work with TMS as well, but only with, you know, college age students or older, so
Right.
you know, much less of an issue. All right, great. Any more questions?
I have a question.
Go for it.
So, I'm fairly naive when it comes to EEG studies, so this could be a naive question. But seeing such high per-trial variability, intra-individual variability, I am wondering if a similar approach, if anyone has tried a sort of dimensionality reduction approach or dynamical systems approach, like some people in the brain-machine interface community have implemented to get at a more maybe a signal that's more stable for a given trial.
Right. Yes. Thank you for that question. And it's, you know, I mentioned this fMRI bold moment or trial to trial measure that we've been using, but there certainly are other labs that are using, for example, a multi-scale entropy measure for oscillatory EEG. That's something that I think in this study we sort of we're more curious about, like that those N400 ERP components, but that's something that I think a direction we could go in the future is another sort of measure of kind of dynamics and how they're impacted by the TMS. So it's a literature I'm a little less familiar with, but I know that there's real interest in developing these kind of variability markers in multiple modalities, and also sort of connecting sort of that fMRI bold variability with, for example, oscillatory dynamics, or with neurochemistry, for example, sort of trying to triangulate all of that. That's definitely something we're curious about, but within the constraints of this, we're sort of more focused on those specific components because we sort of have the prior studies to justify it.
Yeah, of course. Thank you.
All right, and we have a question from Ronnie Lee in the chat. I'm happy to read it. Ronnie, do you want to ask it yourself? I don't want to put you on the spot, so I'll just read it. Um, oh, okay. The question is about how good is sham TMS? So even though you control using sham stimulation in your rTMS studies, is it ever an issue that subjects might still know whether they're receiving actual versus sham stimulation just because they can feel it or hear the clicking?
Yeah, great question. And that's something where, because we're sort of getting our protocols in place right now, we're spending time thinking about that. And I don't know that there's necessarily, from my take, I don't know that there's quite necessarily a standard, but I know that there's sort of thought about the impact of that sham stimulation. And for example, sort of delivering somatosensory stimulation that at least sort of mimics the feel of it on the scalp, or sort of those clicking noises and things like that to try and mimic as close as we can. um, that we're sort of still thinking through the best way of doing that, and looking at the literature for that. So if you have suggestions, that's definitely something that we're very open to at this point, because I agree, it is a real concern.
So if I can just follow up, I actually missed that part of what exactly you said about the sham, but is your sham just normal TMS at a different site, or do you turn the coil, or did
Right. We've
you?
actually talked about just flipping the coil upside down,
Okay. Uh-huh.
because the reason for that, and in other studies, um, what has been used is vertex stimulation. So this is so Nikki Arrington used this in other studies.
Mhm.
But with reading, it gets tough to really. Reading is such a whole brain activity, and so is language, that like, even the vertex. I mean, it's hard to justify that that's really a control region, so to speak, in that it likely will also have consequences for this task. So we sort of thought the best way to handle this was to use the sham stimulation of sort of the inversion of the coil rather than stimulating another region.
Yeah. Another option, just as a suggestion, is sometimes people use S1, the primary somatosensory cortex, which, you know, as a cortex that is specialized for touch, is sort of one of the things that is sort of the least important for other things. I mean, I can see your point, but as long as you have another site and you don't study neuroscientists, so they don't know, you're stimulating here in the brain. I have no idea what each
Right.
brain area is supposed to do, so you're actually, very safe. Um, but yeah, I don't know if you've thought about S1, for example. Yeah.
Yeah. Thank you. Yeah, that's definitely worth thinking about further.
Right, well, great. That was a lot of fun. Thank you so much, Jeff. Here's one more round of applause for you.